Every organism alive has to exchange gases with the environment. If you are an aerobically respiring organism, meaning you make ATP through aerobic cellular respiration, you've got to bring in oxygen, get rid of CO2, but regardless, you have to exchange gases. And very, very small organisms, single-celled organisms, can just do this directly with the environment through diffusion across their cell membrane. But in larger organisms, you can't do that. So because the cells in the inside of my body can't diffuse directly with the environment around me because there are a lot of other cells stacked outside of them, big organisms like me and you have to have specialized structures with lots and lots of surface area to do the exchanging, and then another organ system or circulatory system to move those gases around and distribute them to and from our cell. And one example of a structure like this is a gill, right? Gills allow for a lot of surface area for exchange of oxygen and CO2 with the environment. And we often think about gills as those pharyngeal slitted structures that you find in fish, but really they're any kind of structure that allows for gas exchange in the water. Diffusion happens more effectively across smaller membranes, so this is why you're going to see very, very thin membranes in all these structures for exchange. And also, it's more effective, the exchanges, when you have larger surface area. And very simple organisms like cnidarians have a gastrovascular cavity because they're really only 2 cell layers thick, and as long as they bring oxygenated water into their gastrovascular cavity and have water on the outside, then there's sufficient room for diffusion. Other very tiny organisms can just be flat, and so you don't really have much of a circulatory system in a planarian, for example. They do have a gastrovascular cavity, all right, which water can move in oxygenated and it get delivered to all of these cells. But also, because they're so flat, they actually have a fair amount of surface area for volume, and they're very tiny, and so they can do a lot of exchanging of gases on the outside of their skin too. But in organisms with many cell layers, a circulatory system can help minimize the diffusion distance between the oxygenated fluid and the cells that you're oxygenating. And in order to have a circulatory system, you need some sort of circulatory fluid that can carry gases as well as other things like wastes and food, and a set of vessels that conduct that fluid around the body, and a pump that can create pressure to push the fluid through the vessels. In other words, a heart of some sort. And for some organisms, that circulatory system is open, meaning there is no difference between blood and tissue fluid. It's all one thing. That's called hemolymph. And for instance, insects can just pump that blood up through the heart and then out a series of vessels that just sort of dump the fluid all over the tissues. Closed circulatory system looks more like this. We have a closed circulatory system. Blood is just moved in a circle through the same set of vessels, and it's oxygenated in one place, drops off oxygenated blood elsewhere, and then it comes back to get oxygenated again, passes through the pump. Again, very enclosed, whereas an open circulatory system, you're sucking fluid into the vessels, then dumping them out of the vessels. Whereas open circulatory systems are found, for instance, in insects and other arthropods, closed circulatory systems, which are more efficient at transporting those circulatory fluids to the tissues and the cells, are found in annelids, cephalopods, and vertebrates like us. Ours is called a cardiovascular system. There are 3 main kinds of vessels: arteries, which take blood away from the heart, veins, which take blood back to the heart, and in between those arteries and veins are capillaries, which allow for exchange. So in a way, those are the most important type of vessel because exchange is the whole point, and blood just flows one way in a circle, hence circulatory system. It's important to remember that while we tend to think of arteries as carrying oxygen and veins as carrying deoxygenated blood, that's not necessarily the case. They're defined by the direction of flow, arteries away from the heart, veins back toward the heart, rather than by oxygen content. Though in the systemic circuit, arteries tend to carry oxygenated blood, veins tend to carry deoxygenated blood. And blood comes into the heart, which is the main pump, via a chamber called an atrium. An atrium, like you walk into a building and there's a big atrium, right? In the health professions building in Cincinnati State, the area with the big spiral stairway, that's an atrium, right? That's an entrance room. and then it comes down to a ventricle, which is the big muscular area that pushes the blood out. So blood always enters the heart via an atrium and gets pumped out via a ventricle. Fish have what's called a single circulation, meaning blood leaves the ventricle of the heart and passes through capillaries in the respiratory surface, which are the gills in this case, and then they continue past the gills to drop that oxygen off at the tissues, and then they come back to the heart. So capillary bed at the respiratory surface, then arteries again, then capillary bed, dropping the oxygen off at the tissues, and then back to the heart. Single circulation. And this is what that single circulation looks like, right? We leave the ventricle of the heart, go to the gills, pick up oxygen, continue to the body's tissues, drop off the oxygen, pick up the CO2, and come back to the heart. A double circulation looks a little bit different. You actually go through the heart twice. So you come back deoxygenated to the right side of the heart, and you pump to a set of capillaries in the respiratory surface. In this case, we're talking about a panda, so it's lungs instead of gills. And then we come back to the heart to get another pump on the left side of the heart before we go back out to the systemic circuit to drop the oxygen off at the tissues and pick up the CO2 and come back to the heart. So you get two pumps. And this is important because it gives you extra blood pressure to get all the way through the body. When you're fighting gravity, this is more important. You have to go all the way down to the toes and all the way back up to the heart again. Whereas when you're a fish, gravity's not as big of an issue. You're in a buoyant, supportive environment like water, and so you don't need quite as much pressure to go through. So outside of fish, most vertebrates have two circuits, double circuit. They'll have a systemic circuit that delivers oxygen-rich blood to the tissues, and then either a pulmonary or pulmocutaneous circuit that sends the blood to go pick up oxygen from the respiratory surface. Either the gills or lungs, or in the case of amphibians, also the skin. So let's talk about each of these animals. Amphibians, like frogs, have two circuits, and they have a three-chambered heart. So two atria, so two places where you enter the heart, and one ventricle for pumping blood out. So deoxygenated blood comes back to the right atrium, comes down to the right ventricle, just the one ventricle, and then it gets pushed out through the pulmocutaneous circuit. Again, because this blood is going to pick up oxygen at the lungs, but also at the skin. Amphibians can do gas exchange across their skin. That's why their skin has to be moist most of the time. And then it returns through the left atrium and down to the same ventricle, and it gets pumped out through the systemic circuit to drop off that oxygen to the tissues, pick up CO2, come back to the heart. Now, obviously, you have one ventricle where you're dumping deoxygenated and oxygenated blood and then pumping it out two different places. You're going to get mixing of blood here. And so this is not quite as efficient as a four-chambered heart, which we'll talk about in a moment. But that's not as big of a deal for amphibians, again, because they have this extra respiratory surface in their skin. Reptiles actually look very similar, except that they don't have that skin part. So they have three-chambered heart. They have two atria and one ventricle, but they're a little bit more efficient than amphibians at keeping the deoxygenated and oxygenated blood separate in the ventricle because they have an incomplete septum. So it's still just a single ventricle, but it's a ventricle with two sides, an oxygenated and deoxygenated side, and so there's actually less mixing of the blood than you would have in an amphibian, and so it's a bit more efficient. Reptiles also don't tend to use nearly as much energy as other organisms because they're also, like amphibians, cold-blooded. They don't do nearly as much moving. You see a lot of sitting and waiting from reptiles. But still, pulmonary circuit going to the lungs and back, and systemic circuit going to the tissues and back. Now, mammals and birds have four complete chambers. So they have two atria, just like all of those other organisms, but they also have two distinct ventricles. So that is a complete septum, that interventricular septum that separates entirely the left and right ventricles. So you have deoxygenated blood coming back to the right atrium, to the right ventricle, gets pumped to the lungs through the pulmonary circuit. So pulmonary means lungs. And from the pulmonary circuit, we come back through the pulmonary veins to the left atrium, because we always enter via an atrium. Then we go to the left ventricle, and then that left ventricle pumps this entirely oxygenated blood out to the systemic circuit. Again, this four-chambered heart is much, much more efficient than a three-chambered heart, and that's important because mammals and birds are both endothermic. which means we utilize our metabolism to regulate our internal temperature, and that requires a lot of energy. So we need to not only get in a lot of food, but we need to keep a lot of oxygen pumping to our cells to undergo cellular respiration. And I want you to be broadly familiar with circulation in a mammal, right? So we will start by returning deoxygenated blood to the heart. Again, we start at the right atrium where deoxygenated blood re-enters, both from the inferior vena cava and the superior vena cava, which come from the bottom and the top of the body, right? From your legs versus from your head and arms. We all dump back into the right atrium, deoxygenated, then down to the right ventricle, which pumps out to the pulmonary circuit. And pulmonary goes to the lungs. So we send through pulmonary arteries, because these are leaving the heart, so they're arteries, go to each lung, pick up oxygen, drop off CO2, and now we have oxygenated blood coming back to the heart through veins. Again, I know they're red, I know they're oxygenated, but they are veins in the pulmonary circuit because they're returning to the heart. So these pulmonary veins come from each lung and dump into the left atrium. We always enter via an atrium. From the left atrium, we go down to the left ventricle and then get pumped out the aorta. And then it branches to go to the top part of the body or the bottom part of the body. There's lots of branching from there, but this is the systemic circuit, and we'll drop off oxygen at the tissues, pick up CO2, and then come back to the heart. And if you look closer, you can see these chambers, right? Right atrium, right ventricle, left atrium, left ventricle. You can also see that there are valves, atrioventricular valves between the ventricles and the atria, so that when this ventricle pumps, it doesn't just pump the blood right back up into the atria. Right? These valves prevent backflow. They close when the ventricle pumps so that it goes, for instance, out the aorta instead of back up into the left atrium. Or on the right side, it doesn't go back to the right atrium. It goes from the right ventricle out the pulmonary trunk into the pulmonary circuit. And your heart will contract and relax and contract and relax in a cycle called the cardiac cycle that includes contraction, which is called systole, and relaxation, which is called diastole. Relaxation when you're refilling the ventricles. So systole, contraction, diastole, relaxation. This is why your blood pressure has a systolic blood pressure and a diastolic blood pressure. Because after contraction, right, the blood pressure in your vessels is much higher than after relaxation, right? It goes in waves. And so the systole is the highest your blood pressure will possibly be, right? Systolic right after contraction. Your blood pressure right after diastole or relaxation is the lowest it's going to be. And this heart rate is set by your pacemaker or your sinoatrial node. And this just constantly sends an electrical signal to get your heart to beat at a regular rhythm. It can be adjusted in terms of its rate by your brain so that when you're exercising it goes up and when you're relaxing it goes down. But your sinoatrial node is necessary. And now if it's not working, you can get an artificial pacemaker put in. And if you want to see what someone's cardiac cycle is looking like, you can record that aggregate electrical activity through an electrocardiogram, which is sometimes called an ECG or an EKG. Same thing. And if you look at the structure of these vessels, right, we leave the heart via an artery, right? And arteries have three layers: endothelium in the inner layer, some muscle in the middle, and connective tissue in the outer layer. But a lot of muscle and elastic tissue there, because arteries, especially the ones right by the heart, have to deal with huge swings in pressure during systole and then snap back during diastole. Right? And then we go into smaller arteries called arterioles, and then we go into capillaries, which look very different structurally. They're tiny because diffusion happens more effectively across a thin membrane, so they are literally a single cell thick, and they're so narrow in diameter that only a single red blood cell can pass through at a time. This allows for a ton of surface area for exchange. It's fantastic. And then we drop off oxygen, pick up CO2, at least in the systemic circuit. It's the reverse in the pulmonary circuit. And our deoxygenated blood goes through these venules into larger and larger veins, which have the same three layers as arteries, although thinner muscle, less elastic tissue, and those will dump back into your heart. And as I mentioned, your blood pressure comes in spikes and dips. So spikes during systole, dips during diastole, and the difference between those two is your pulse pressure. So when you're feeling your pulse, right, what you're really feeling is the difference in pressure between relaxation pressure, diastolic pressure, and contraction pressure, or systolic pressure. And so this is a good way to get an estimate of your of your heart rate, I should say, because when your heart pumps, each pump you get a pulse or a jump in blood pressure.